Okay, well, I will go over, hopefully, the final installment of this particular review, which is nuclear factor kappa B, its role in inflammation. So if you haven't seen any of the three previous videos on it, please watch those first. And granted, that would be over 3 hours worth of content. But if you do find the time, I would highly recommend going over those videos before we move on to this video. So nuclear factor kappa B in inflammatory disease, and remember, nuclear factor kappa B, this is a multiple subunit protein complex. So the canonical version of it is utilizing a rel-A, which is one protein, and a closely related protein known as P50, which comes from P105. Wait, is it P105? Yeah, it comes from a previous protein, which I believe is P105, and it gets proteolytically cleaved into P50. So those two subunits will come together to form nuclear factor kappa B, which then usually has an inhibitor protein that also will associate with it. And certain signals in the cell will then cause the phosphorylation of the inhibitor protein, which then makes that inhibitor protein pop off and then gets degraded. So when it comes to your newly activated nuclear factor kappa B, that then goes to the nucleus where it affects transcription, which is why it's called a nuclear factor, right? So nuclear factor kappa B has been implicated in the pathogenesis of a number of inflammatory diseases, such as rheumatoid arthritis, inflammatory bowel syndrome, multiple sclerosis, uh, I can never pronounce this one, God dang it, atherosclerosis, systemic lupus, erythematosis, and I do not know what this one is. I'd assume it's something red. Yeah, so basically causing redness, but also which is associated with lupus. So let's see if there is a Wikipedia article so that I don't. Wikipedia should just always be the first thing. So when someone has lupus, you can see here that they are very frequently get rashes, and I really should look up more about what lupus does, but just know that this is one of the side effects or one of the symptoms of it. So you can get this. It is also implicated in type 1 diabetes, chronic obstructive pulmonary disease, and asthma. So these all seem to be associated with, to a degree, with nuclear factor kappa B being dysregulated in some way. In response to different cellular stimuli, nuclear factor kappa B plays a complex role in different cell types and in different diseases state. Different diseases states? I think that is probably a typo. Regardless, let's keep on going. So in rheumatoid arthritis, this is an autoimmune and inflammatory disease which is characterized by the immune cell infiltration into the synovium, the synovium being the fluid that is within your joints. And this is associated with chronic inflammation and destruction of cartilage and bone. So a major inflammatory mediator of rheumatoid arthritis is nuclear factor kappa B, which has been demonstrated in studies using both animal models and human patients. So that's what we like to see when we have in vivo examples of how they're playing a role. So, for example, several studies have detected nuclear factor kappa B activation in synovial tissues of rheumatoid arthritis patients. So, in mouse collagen-induced arthritis, nuclear factor kappa B activation in synovial tissue precedes the development. So this is happening first. It precedes the development of clinical symptoms and increases along with disease progression. So collagen-induced arthritis. Let me just see if I have this correctly. This sounds like they inject collagen. So is a condition induced in mice to study rheumatoid arthritis, and CIA is induced in mice by injecting them with an emulsion of, so complete Freud's adjuvant and type 2 collagen. So adjuvants are things that we add to, say, vaccines, or they are just chemicals that we add to an active ingredient of some form, which makes the response to that active ingredient greater. So adjuvants, they play a role in, say, pesticides, right? So some pesticides will have their the active ingredient chemical, but sometimes they will also have adjuvants to make the plants or the pests more susceptible to that active ingredient. So when it comes to vaccines, we have adjuvants that help the immune system recognize the active or the main ingredient, which is oftentimes the antigen. And so this is how you will develop an immune response against the antigen. Now, when it comes to causing rheumatoid arthritis in mice, to study them as a model organism, they will inject type 2 collagen, which, collagen is one of the proteins that holds your body together. They will inject this collagen, which normally the body should not develop a huge immune response to. But then they add an adjuvant, and in this case, it's the complete Freud's adjuvant, and this makes the immune system hyperactive and develops antibodies and develops an immune response against collagen. Now, think of this. After you have developed an autoimmune response to collagen, then that immune system can turn around and then attack the collagen that is within your own body. And in the case of this is happening to mice. So in mice that receive this collagen-induced arthritis, this nuclear factor kappa B activation is, it's activated in the synovial tissue, and it's activated before you can see the rheumatoid arthritis symptoms. And as the rheumatoid arthritis progresses worse and worse, the nuclear factor kappa B expression goes up. Or I shouldn't say expression, but activation goes up. So nuclear factor kappa B activation has also been associated with rat arthritis induced by different agents such as pristine and streptococcal cell wall. So pristine, I presume this is just a chemical that they inject, and pristine injection. So let's chemical. So I do not know exactly what pristine is. Wait, let me look up pristine arthritis. Oh, okay, so this must be a typo. This should be pristine, not pristine, because it looks like, yeah, pristine. So let's look up pristine and see what exactly it is. So this is pristine. It's just it's a hydrocarbon. It's a saturated terpenoid alkane. So terpenoids are chemicals that come out of isoprenoids. Regardless, you don't need to worry too much about that. So just know that this is what pristine is, and then by injecting this into an organism, you can induce a form of arthritis. So we can induce arthritis with pristine, and we can cause arthritis through streptococcal cell wall, which streptococcus, this is one of the pathogens, right? And if we take the cell wall out of streptococcus, we can activate, how to phrase this, we can activate an immune response, and that immune response can also cause arthritis. So keep in mind, sometimes you do not want to make antibodies against something. Sometimes you do not want to have an immune response against something, because then that immune response can damage yourself. So in the context of, say, a global pandemic, when we have this novel coronavirus that is circulating around, we might not want to run that experiment on people to see whether or not they develop autoimmune diseases. Because there are a lot of proteins in the body that um, that could accidentally be mimicked by a virus. So, continuing, similarly, nuclear factor kappa B activation in rats by intra-articular transfer of an adenoviral vector encoding the IKK beta. This induces synovial inflammation and clinical signs of arthritis. So, basically, if you frequently we will use adenoviruses or other types of lentiviruses would be another form, which I believe are actually from HIV. But regardless, you can take viruses and you can genetically engineer the viruses so that they can have genes of interest in them. And so when you insert this virus into a host organism, the virus is then going to inject its genetic components into the cells that they infect. And we design these viruses so that they can't multiply, they can't duplicate, but they can still inject the genetic material into an organism. And so that injection of genetic material can be used to genetically engineer or add genes into organisms that wouldn't otherwise have that particular type of gene. So in this particular case, they are giving IKK beta, which IKK beta is the, let me go back to, they had a really good figure. And, oh, here, wait. Yeah, so IKK beta is, I should presume would be one of the subunits of this IKK. But IKK will attack or add phosphates to this I kappa B, and by adding phosphates to this I kappa B, it'll pop off, which activates nuclear factor kappa B. So what they have done here is they have added, or they've made an adenovirus which encodes IKK beta, and basically, rather than it being a regulated expression of IKK beta, you basically have constitutive or much more expression of IKK beta. And so by doing this, you are now activating nuclear factor kappa B by removing the inhibitor from it. So, and if you do this, you see synovial inflammation or inflammation of the cells in the synovial fluid, excuse me, and you will also see clinical signs of arthritis. Whereas intra-articular transfer of a dominant negative IKK beta mutant suppresses adjuvant-induced arthritis. So I don't know exactly. A lot of big words there, and I can't parse it exactly. So let's look at what intra-articular transfer is. And I just need to wiki. Okay, so that's a joint injection. Why don't they just call it a joint injection? Jeez. So, so when they do a joint injection of a dominant negative IKK beta mutant, so I don't know what dominant negative means. Mutation whose gene product is adversely affects the normal wild type gene product. Okay, so when they add a dominant negative version of IKK beta, this is a mutant that does not work, or doesn't work the same way. And in fact, by introducing it, it makes the wild type or the normal gene work worse. So by adding the dominant negative IKKB mutant, it will suppress the adjuvant-induced arthritis, which, remember, the adjuvant-induced arthritis is usually from injecting collagen with some other adjuvant, which makes the immune system go crazy. So in line with these, and if you do this, you end up reducing the arthritis in the organism. So in line with these findings, mice with myeloid cell specific deficiency of A20, okay, we're going to have to go back to that. What was A20 again? You know what? They don't have it here. Let's see if there's one down here, and no, there isn't. So we'll just keep on going. Hopefully I'll be able to parse this. So in line with these findings, mice with the myeloid cell-specific deficiency of A20, which is a D-ubiquitinase, which negatively regulates nuclear factor kappa B signaling. So if you do not have A20, you end up getting too much, or you end up positively regulating nuclear factor kappa B. If you remove this A20 from the myeloid cells, you end up with spontaneously developing polyarthritis with typical features of rheumatoid arthritis. So if you get rid of one of the proteins that will suppress nuclear factor kappa B, you end up getting arthritis. So finally, nuclear factor kappa B inhibition by decoy oligonucleotides or the IKK inhibitor known as BMS 345541, these will ameliorate adjuvant-induced arthritis. So if we can inhibit nuclear factor kappa B through multiple mechanisms, one through the inhibitor, one through the decoy oligonucleotides, and oligonucleotides, these will be small bits of genetic information that can help basically they can affect translation or transcription in different types of ways, but if you do either of those, you can downregulate nuclear factor kappa B, and by downregulating nuclear factor kappa B, you end up removing the negative consequences of this adjuvant-induced arthritis. So, the pathogenesis of rheumatoid arthritis involves a variety of cell types, including innate immune cells such as the monocytes, macrophages, T cells, B cells, and synovial fibroblasts. What are the synovial fibroblasts again? They are found in the synovial. Oh wait, let's use Wikipedia. Oh, these are the fibroblast-like synoviocytes. Okay, so yeah, these are just cells that exist in the synovium. So, nuclear factor kappa B mediates the induction of pro-inflammatory cytokines such as TNF-alpha, IL-1, and IL-6 in the monocytes and the macrophages. So many of these cytokines are capable of activating NF-kappa B in innate immune cells and fibroblasts, thereby inducing the expression of additional inflammatory cytokines and chemokines. So remember, this is often this is the positive feedback loop. Pro-inflammatory cytokines are creating or causing the induction of more pro-inflammatory cytokines. So this is leading to further recruitment of inflammatory immune cells and dissemination of inflammation. The canonical and non-canonical nuclear factor kappa B pathways also mediate rank ligand-induced differentiation. So let's monocytes and macrophages into the bone-resorbing osteoclast. So let's look this up real quick. So what is the rank ligand-induced differentiation? And let's look up Wikipedia. So is this, yeah, so rank L, so receptor activated of nuclear factor kappa B ligand, also known as rank L. This is also known as tumor necrosis factor ligand superfamily member 11, or TNF-related activation-induced cytokine, also known as trans, also known as osteoprotogerin ligand, or OPGL, and also known as osteoclast differentiation factor, ODF. So here we have 8 names. I'm being a little hyperbolic, but here we have 8 freaking names for the same freaking protein. It's absolutely ridiculous biology. Just get your crap together. Regardless, so this is the rank L or rank ligand, and it is a type 2 membrane protein, so it exists in the membrane of the cell and is a member of the tumor necrosis factor superfamily. So rank L has been identified to affect the immune system and control bone regeneration and remodeling. So rank L is an apoptosis regulator gene, a binding partner of osteoprotogerin, which is a ligand for the receptor rank, and it controls cell proliferation by modifying protein levels of these things. So When it comes to the canonical and non-canonical nuclear factor kappa B pathways, they also will mediate these rank ligand-induced differentiation of monocytes and macrophages into bone-resorbing osteoclasts. So I didn't actually know that. So these white blood cells will go in to will differentiate into osteoclasts. And if you deregulate this process, it contributes to inflammatory bone loss, which is associated with rheumatoid arthritis. So I did not. So these are large multinucleate bone cells, and they absorb bone tissue during growth and healing. Let's read this real quick. So osteoclast meaning bone, and clastos meaning broken, and it's a type of bone cell that breaks down bone tissue, and the function is critical for the maintenance, repair, and remodeling of bones in the vertebral skeleton. And the osteoclast disassemble and digest the composite of hydrated protein and mineral at the molecular level by secreting acids and collagenases, which is a process known as bone resorption, and this process also helps regulate the levels of blood calcium. So if you could imagine, if you create a bone, you don't want your bone, you might not necessarily want that bone to always exist. Maybe you need to go through a specific period of time where you are destroying little bits of bone. That way you can heal properly, or let's say a piece of bone breaks off and then it just moves a little bit. Well, maybe you want to destroy that little piece of bone because it's no longer where it's supposed to be. So this is what the osteoclasts are doing, is they are attacking the bone in order to liberate either space or the minerals or the nutrients that are associated with that bone. So if you have a deregulated nuclear factor kappa B, you might produce too many osteoclasts which come from the monocytes and macrophages. And if you do that, you then end up with bone mineral loss, which is no bueno. So although the different subsets of T cells, T helper 17 cells, are particularly important for the pathogenesis of rheumatoid, among the different subsets of T cells, the T helper 17 cells are particularly important for the pathogenesis of rheumatoid arthritis. As described above, nuclear factor kappa B promotes T helper 17 differentiation both indirectly through the induction of inflammatory cytokines, such as IL-1, IL-6, and IL-23, in innate immune cells, and it also directly regulates the T helper 17 lineage transcription factors in the T cells. So deregulated activation of nuclear factor kappa B also contributes to aberrant survival of self-reactive B cells and production of autoantibodies, that contribute to the pathogenesis of rheumatoid arthritis. So, as we said earlier, if you deregulate nuclear factor kappa B, you will have the aberrant survival of self-reactive B cells. So, the self-reactive means B cells would be B cells that attack your own body, right? And you would hope that these would die very quickly, because of dysregulated nuclear factor kappa B, these white blood cells, rather than just dying, they end up living, existing, and I would presume duplicating faster or more than what you would want them to, and this could contribute to rheumatoid arthritis. So in particular, RA patients often displayed elevated serum levels of B-cell activating factor belonging to the tumor necrosis factor family, associated with deregulated activation of the non-canonical nuclear factor kappa B. So in rheumatoid arthritis patients, they have high serum levels of the B-cell activating factor. So, and through some logic, if you have too much of this factor floating around, you're going to have too many B cells floating around. So, therefore, nuclear factor kappa B mediates the pathogenesis of rheumatoid arthritis by functioning in different cell types. So, inflammatory bowel disease. So, inflammatory bowel disease, including Crohn's disease and ulcerative colitis, are chronic inflammatory disorders of the gastrointestinal tract thought to result from inappropriate inflammatory responses to intestinal microbes. So remember, you have this microbiome that exists in your large intestine, well, your entire intestine, I would presume. And these play a very important role in nutrient processing, signaling and talking with the body, protecting your body from other pathogens coming in. But these microbes are always secreting different antigens and chemicals and just a whole bunch of stuff into the gut in order to help maintain homeostasis, right? So if somehow your immune system doesn't recognize these beneficial bacteria correctly, you could end up having an inappropriate inflammatory response. And so this can cause things such as Crohn's disease, ulcerative colitis, and chronic inflammation, or chronic inflammatory disorders within the intestines. So the pathogenesis of irritable bowel syndrome involves multiple cell types of the mucosal immune system, which including intestinal epithelial cells, innate immune cells such as macrophages and neutrophils, T cells, and innate lymphoid cells. So strong evidence suggests the involvement of nuclear factor Kappa B in the pathogenesis of irritable bowel syndrome. If you have constitutive nuclear factor Kappa B activation, it has been found in inflamed colonic tissues of the, how do I phrase that? The constitutive nuclear factor Kappa B activation, this has been found in the inflamed colonic tissue of irritable bowel syndrome. Wait, what is it called? Irritable or inflammatory bowel disease in patients. Sorry, I was saying irritable bowel syndrome, but I meant to say inflammatory bowel disease. So furthermore, genetic mutations of nuclear factor Kappa B stimulating immune receptors, such as NOD2 and NF-kappa B target genes, such as the IL-12 and IL-23, these are associated with human inflammatory bowel diseases. So yeah, if you have genetic mutations in the receptors of some of the chemicals that are produced when nuclear factor Kappa B is activated, such as interleukin 12 and interleukin 13, if some of the receptors are mutated so that they are more easily activated by interleukin 12 and or interleukin 23, you can see some irritable bowel syndrome, not irritable bowel syndrome, but inflammatory bowel diseases. You can see higher rates of that in people who have mutations in the receptors of these chemicals, which come from or are upregulated by nuclear factor Kappa B. So polymorphisms and mutations of the NFKB1 gene, which encode the I-kappa B-like molecule P105 and its processing product P50, these have also been associated with inflammatory bowel diseases. So if you have a mutation in the nuclear factor Kappa B1 gene, which remember, this nuclear factor Kappa B1 gene encodes for P105, which is processed into P50, which is a part of the canonical nuclear factor Kappa B molecule. If you have these mutations in this gene, you could have specific types of mutations that are associated with irritable bowel syndrome. So these few nucleotide mutations that are seen in the progenitor gene, then this might result in propensities of developing a disease later in life. So this is one of the reasons why, genetic counseling might become a very big thing soon, is because by understanding people's genetics and by understanding the polymorphisms of the genes inside people, we will have a better understanding on what type of diseases they will get later in life. So these genetic alterations appear to inhibit nuclear factor Kappa B1 gene expression or alter the stability and function of the protein products. Consistently, The mice carrying a knock-in mutation, so the opposite of knockout, a knock-in mutation in the NFKB1 gene to block generation of P105, these will spontaneously develop intestinal inflammation with IBD-like features. And a number of other animal model studies have also demonstrated that genetic deficiency in negative regulators of the canonical NF-kappa B pathways, such as D-ubiquitinases, CYLD, and A20, and remember A20 was the one that if we got rid of it could also cause rheumatoid arthritis-like problems. If you remove this, how would I say that? If you create genetic deficiencies of these negative regulators, such as CYLD and A20, you can promote colonic inflammation. So, or the IBD. So in line with these findings, decoy oligonucleotides that target the DNA binding activity of nuclear factor kappa B. Oh, sorry, I must have misspoke earlier. So using decoy oligonucleotides would be little bits of DNA that look like the binding sequence or the target sequence of nuclear factor kappa B, and by introducing a bunch of these little oligonucleotides into the system, the nuclear factor kappa B gets sidetracked. It's not able to bind to the DNA within the nucleus. They end up binding to these decoy oligonucleotides, which prevent them from functioning or acting upon the transcription of the DNA, if that makes sense. I apologize. There's many different ways that you could have decoy oligonucleotides, and this is the one that they were talking about earlier. So, if you add these decoy oligonucleotides and they lower the activity of nuclear factor kappa B, this can ameliorate the colitis, which is induced by trinitrobenzene sulfonic acid and dextrin sulfate sodium. Big words. Just know that if you add these, you can cause colitis, and by adding the decoy oligonucleotides, you can heal or prevent some of that colitis, and we presume that effect is from targeting nuclear factor kappa B. So, deletion of the IKK beta in myeloid cells also inhibits experimental colitis and colitis-associated cancer. Interesting. So these findings are consistent with the role of nuclear factor kappa B in mediating induction of pro-inflammatory cytokines in innate immune cells and the differentiation of Th1 and Th17 subsets of inflammatory T cells. So, in contrast to its pro-inflammatory role in myeloid cells, nuclear factor kappa B has a protective role in intestinal epithelial cells, where it is required for maintaining epithelial integrity and intestinal immune homeostasis. Yeah, so conditional deletions of NEMO, IKK beta, both IKK alpha and IKK beta, and intestinal epithelial cells, these cause spontaneous development of chronic intestinal inflammation in mice. Thus, aberrant activation of nuclear factor kappa B or its genetic deficiency may both contribute to pathogenesis of IBD, with its functions differing between innate immune cells and epithelial cells. So, here we're saying that nuclear factor kappa B, not only can it have a pro-inflammatory effect in some cells, it can have an anti-inflammatory effect in a different subset of cells. So what they're saying is this is all supposed to be a tightly regulated process with both positive and negative feedback loops, and if either of those get screwed up, you can still end up with these diseases. So, continuing. Multiple sclerosis is an inflammatory disease of the central nervous system, also called CNS, and it's generally considered to be an autoimmune disease involving the pathogenic actions of the central nervous system specific CD4 positive T cells, particularly the T helper 1 and the T helper 17 cells. The involvement of nuclear factor kappa B signaling pathway in multiple sclerosis has been suggested by genome-wide association studies And these studies have identified a number of nuclear factor kappa B related factors as susceptibility candidates. And these are REL-A, I kappa B alpha, I kappa B Z, or zeta. Yeah, I presume that's zeta. NIK, BCL10, and MALT1. So remember, these are all proteins that are associated with the proper regulation of nuclear factor kappa B. And so in people with multiple sclerosis, they seem to have found candidates associated with multiple sclerosis, and quite a few of them are associated with nuclear factor kappa B. So consistently, both the canonical and non-canonical nuclear factor kappa B pathways play an important role in the pathogenesis of EAE, which is experimentally acquired encephalitis. Wait, let me. No. Yeah, okay. Experimental autoimmune encephalomyelitis. Okay, so this is inflammation of the nervous system that it has been acquired through an experimental injection of some form, which causes an autoimmune disease where the own immune system gets attacked. So that's what EAE stands for. So it's widely used, and this is a widely used animal model of multiple sclerosis, which is involving the immunization of mice with peptides derived from CNS proteins, such as myelin, oligodendrocyte glycoprotein. So a few decades ago, there was an issue with some meat packing workers where a lot of them were getting, I think, multiple sclerosis, or at least inflammation of their nervous system, because they were breathing in particulate matter of the nervous system of the pigs that they were processing. And so basically the immune system ended up mounting a response against the myelin of the nervous system tissue of the pigs after it was breathed into the humans, and then that caused an autoimmune disease against the myelin sheaths of their own nervous system, which then results in multiple sclerosis-like diseases. So this is something that doesn't just happen experimentally, this happens in the real world as well. So T-cell specific deletions of IKK beta or oral administration of an IKK beta inhibitor, also known as PS1145, this renders mice refractory to EAE induction. It renders the mice resistant to EAE induction. So if you remove IKK beta, or if you inhibit IKK beta, you are then inhibiting nuclear factor kappa B from functioning or activating properly. And if you do this, some of the mice are more resistant to the EAE induction, or they are more resistant to this autoimmune disease. So genetic deficiency in IKK upstream signaling factors of the TCR pathway, including KARMA1 and MALT1, also ameliorate EAE induction. So remember, just know that KARMA1 and MALT1, these are upstream of IKK, and so if you affect these factors, it also ameliorates EAE. And I presume that KARMA1 and MALT1 positively influence IKK. So the canonical nuclear factor kappa B members REL-A, CREL, and CREL mediate expression of the T helper 17 lineage transcription factor ROR gamma T, and thereby promotes T helper 17 differentiation. An important role of non-canonical NF-kappa B pathways in EAE regulation has been demonstrated using mutant mice which lacking the kinase NIK or expressing processing defective P100 mutant. So remember, P100, this is one of the non-canonical subunits of nuclear factor kappa B, so it's not the one that is normally expressed. So if you remove, or if you express a P100 that cannot be processed into P52, which P52 is a part of the non-canonical nuclear factor kappa B subunit or complex, then if you cannot get this to happen, you end up screwing up the ability for the, or I shouldn't say, you end up adding resistance to the mice against experimentally autoimmune encephalomyelitis. Oh man, EAE. So let's see. Regarding the latter, the non-canonical NF-kappa B member P52, in synergy with CREL, this mediates the expression of inflammatory cytokine GM-CSF in the T helper 17 cells. So, and I presume the kinase NIK plays a role in the processing of P100. So if you don't quote me on that, there's way too much to remember, but I believe if you remove NIK or you lack the kinase NIK, you then lack the ability to process P100 into P52. So affecting both either NIK or P100 directly can influence whether or not P52 gets created. And if P52 can't get created, then you can't create this non-canonical NF-kappa B, and therefore you cannot create proper levels of this GM CSF. So, in addition to its function in regulating the differentiation and effector functions of T cells, NF-kappa B regulates EAE through the action of innate immune cells, constitutive activation of NF-kappa B in myeloid cells as a result of I-kappa B alpha deletion using the LYSM-CRE system. Don't worry too much about this. Just know that there is a system by which you can deactivate genes later in life. This would be one of them. This causes more severe central nervous system inflammation. So, let's say you use this LYSM-CRE system to destroy or deactivate this inhibitor I-kappa B alpha. By causing the I-kappa B alpha to be deleted, this causes nuclear factor kappa B to be hyperexpressed in these myeloid cells. So they might be using this to target just the myeloid cells. And let's look this up just to, yeah, so these are a type of, these are the blood or the bone marrow tissues. So yeah, if you can do that, you can cause more severe central nervous system inflammation. And clinical scores in myelin oligodendrocyte glycoprotein-induced EAE. Whereas myeloid cell-specific deletions of IKK beta inhibits the EAE induction. So remember, this is the inhibitor. If you remove the inhibitor, then you end up causing more severe CNS inflammation. And this IKK beta is something that destroys the inhibitor. So if you destroy the destroyer, or if you get rid of the destroyer of the inhibitor, then you end up creating resistance to EAE induction, which would be good. So, and this is associated with impaired generation of inflammatory T helper 1 and T helper 17 cells. So, nuclear factor kappa B also functions in the central nervous system to regulate neuroinflammation, since conditional deletions of NEMO or IKK beta using the NES-CRE, which is specific for neuronal cells, including neurons, astrocytes, and oligodendrocytes. This partially inhibits EAE induction. So rather than using this, what's it called, LYSM-CRE system, they use this NES-CRE system, which is specific to the nervous tissue. And by using this system, they are able to turn off the, or cause conditional deletion of NEMO or IKK. And by doing this, we can have partial inhibition of EAE induction. So we can, if we just focus on the neurons, we can have some positive effect of that. So furthermore, the nuclear factor kappa B inhibition of astrocytes through transgenic expression of a degradation-resistant form of IKB alpha. This inhibits inflammatory cytokine expression and reduces the disease severity in EAE. So, oh, and one of the things that I want to just pause and take a mention here, experimentally autoimmune encephalomyelitis. This is something that, at least in mouse models, we use coronaviruses to cause EAE in some mice. So one of the things that I'm a little concerned about right now is that this new coronavirus pandemic might introduce the ability for inflammation of the brain into a lot of people that would have otherwise not had it. And because of this, I am worried that a few months to a few years down the line, we are going to see a lot of very serious nervous tissue damage that is associated with inflammation that was caused by the SARS-CoV-2 coronavirus. And we will not find this out fully until, a few years to a decade. So that's just one of the downsides with the new pandemic. So, atherosclerosis. So atherosclerosis is a progressive and inflammatory disorder of the arterial wall characterized by accumulation of low-density lipoprotein particles and immune cells in the subendothelial space. So these are, might as well get a photo, but you've probably seen the photos of people with high cholesterol, and you look up a photo of their, of their blood vessels, and you see all this yellow fatty tissue that is just underneath the top layer. That is what they're referring to when they refer to atherosclerosis. And Wikipedia is not the first option, and I hate that. So here we have an example of a blood vessel. So here we have the epithelial tissues, and here you can see atherosclerosis here. So this is the buildup of the LDL cholesterol, or just fat. And this can get oxidized and become a pro-inflammatory environment, but just, that's what we are referring to. So it's progressive and inflammatory disorder of the arterial wall, and let's see, the pathogenesis of atherosclerosis involves different cell types, including the endothelial cells, the monocytes, and the T cells, and monocytes are just a type of white blood cell. So it is generally thought that the disease initiation involves activation of endothelial cells to express chemotactic factors. So these would be different chemicals that convince certain cell types around them to move into the area. So it was generally thought, or it is generally thought, that the disease initiation involves the activation of the endothelial cells to express these chemotactic factors and cell adhesion molecules that mediate recruitment of blood monocytes into the arterial intima. And then after these monocytes, through these chemotactic factors, are brought into this arterial intima, they then differentiate into macrophages, and following uptake of LDL particles, eventually become lipid-laden foam cells. So basically, these monocytes differentiate into macrophages, and then these macrophages start eating these LDL particles. And by eating these LDL particles, you end up accumulating a lot of fat within them. And by accumulating all this fat, they end up becoming these lipid-laden foam cells. And these are involved in atherosclerotic plaque formation. So I presume that these are stuck here partly in response to the cell adhesion molecules that are being expressed by the epithelial cells. So nuclear factor kappa B regulates the expression of a large array of genes involved in the different aspects of atherosclerotic pathogenesis. So in vascular endothelial cells, the NF-kappa B mediates induction of the pro-inflammatory cytokines, the chemotactic factors, which are these are the factors that will theoretically bring those monocytes into that area of the cell wall, or area of the blood vessel, and adhesion molecules. So nuclear factor kappa B mediates the inflammation it mediates the chemotactic factors, and it mediates the adhesion molecules that keep the cells in place. This is thereby promoting monocyte recruitment and disease progression. So conditional deletions of NEMO, or transgenic expression of degradation-resistant I-kappa B alpha in endothelial cells. This inhibits chemokine expression and monocyte recruitment, coupled with reduced disease severity of atherosclerosis. So, if we add the inhibitor, we seem to have a better outcome with the severity of atherosclerosis. So, in the ApoE deficient mice with the and this is happening in the ApoE deficient mice with the cholesterol-rich diet. So, NF-kappa B also functions in myeloid cells to promote inflammatory gene expression and conversion of macrophages into foam cells. Transgenic expression of a non-degradable I-kappa B alpha, this is the inhibitor, in macrophages reduces the lipid loading and foam cell formation. So if you add an inhibitor that can't be degraded easily, it will protect more of the, or I should say will sequester more of the nuclear factor kappa Bs in the cell, and when you sequester it, then that is helping reduce the ability of these macrophages to turn into the foam cells. So, whereas myeloid-specific I-kappa B alpha deletion sensitizes atherosclerosis development in LDL receptor deficient mice. Myeloid-specific. Okay, so in line with these findings, myeloid cell-specific deletions of IKK beta reduces atherosclerotic lesion areas in LDL receptor deficient mice, which are fed a high-fat diet, which is associated, and this would be the LDL receptor deficient mice. These are probably a model organism to study atherosclerosis because it is very easy to induce atherosclerosis in these particular mutant mice. So, and this is associated with attenuated activities of macrophages in inflammatory gene expression, adhesion migration, and lipid uptake. Surprisingly, however, an earlier study suggests that deletion of IKK beta in myeloid cells increases atherosclerotic lesions in LDL receptor deficient mice. The reason for such discrepancy is unclear, although it could be due to differential expression of experimental approaches used in these two different studies. Good. So, the concluding remarks. So it is now well accepted that nuclear factor kappa B serves as a central inflammatory mediator that responds to a large variety of immune receptors. Since deregulated nuclear factor kappa B activation is involved in various inflammatory diseases, targeting the nuclear factor kappa B signaling pathway represents an attractive approach for anti-inflammatory therapies. Several categories of inhibitors have been developed to block different steps of the nuclear factor kappa B signaling, and an increasing number of selective IKK inhibitors has been diagnosed or has been designed to block the catalytic activity of IKK and prevent IKB alpha phosphorylation. And what I would presume would be a good reason for developing a bunch of different IKK inhibitors is that we could develop inhibitors that are specific to a particular cell type. So if we want to, let's say we want to downregulate we want to downregulate nuclear factor kappa B in a particular cell type, but we want to leave the other cell types untouched. Well, if we just add an inhibitor, then that could negatively affect all of the tissues of the body if we're deregulating all the tissues in the body. But if we want to target a specific tissue type, especially a tissue that is undergoing pathogenesis at that particular point in time, we might want to use an inhibitor that is specific to a specific cell type. So that's one of the benefits of developing or having, what would the word be, robustness, or what's the word in redundancy. So that's one of the reasons why having redundancy in our pharmaceuticals is really important, because then we can have a wide variety of different chemicals that we can choose from. So anyways, some well-known anti-inflammatory drugs such as aspirin and salicylate also have the ability to inhibit IKK. Interesting. So yeah, that is really interesting. Proteasome inhibitors such as Velcade, also known as bortezomib, and PS341, and lactocytocin, which blocks I-kappa B alpha degradation in the proteasome. We also have inhibitors that block nuclear translocation of different NF-kappa B subunits, such as tacrolimus and I-kappa B alpha superrepressor. We have drugs that inhibit DNA binding activity of nuclear factor kappa B, such as the glucocorticoids and PPAR agonists. However, while significant progress has been made in designing approaches to inhibit nuclear factor kappa B, complexities exist for the development of clinically available nuclear factor kappa B based drugs. And although nuclear factor kappa B inhibition could be beneficial in treating inflammatory diseases, there are obvious questions regarding the balance between efficacy and safety, since nuclear factor kappa B function is also required for maintaining normal immune responses and cell survival. So accumulating studies suggest that global inhibition of nuclear factor kappa B signaling may cause severe side effects. Therefore, better understanding of the mechanisms underlying the pathological activation of nuclear factor kappa B in individual diseases. This is crucial for designing more specific and effective therapeutic agents for the treatment of inflammatory diseases. So here they have shown, so in this concluding remarks, they've gone over multiple different types of ways of influencing nuclear factor kappa B activation by attacking different parts of the pathway. So here they have described it, where here we have our cell membrane. These little green spheres are the diverse stimuli, and then you have these receptors on the surfaces of your cells that can activate nuclear factor kappa B, right? So one of the things that we can do is we can develop drugs that will prevent these stimuli from acting upon the receptors. We can develop IKK inhibitors, which will prevent IKK from phosphorylating I-kappa B alpha effectively, and that's one way to inhibit this entire NF-kappa B pathway. We can develop proteasome inhibitors. So remember, you have your I-kappa B alpha, which inhibits the nuclear factor kappa B, and this gets phosphorylated. And here they have a phosphate, but really there should be multiple phosphates on it. Now, this ends up popping off and gets ubiquitinated with all these UBs on it, and then this gets targeted to go into the proteasome, where it's destroyed. We can use proteasome inhibitors, which prevents this from getting destroyed, and I would presume that by preventing this from being destroyed, there might be a semi-reversible process that can have it inhibit more nuclear factor kappa B. Another drug that we can use is a subset of drugs that prevent this nuclear factor kappa B from getting into the DNA. And so if you can inhibit this from translocating into the nucleus, then that's one way to inhibit its effects. Now, another thing that we can do is using DNA binding inhibitors. And so, I mean, I think of an example might be these decoy oligonucleotides that might be able to bind to the nuclear factor kappa B to prevent it from binding properly to the DNA, right? So that would be one example of another type of drug. So, with that, yeah, I think with all of this, I am going to head out. So we are done and just under an hour, so great. I'm glad that I can finally get this review behind me. Yeah, I'm really surprised that there was a few typos in here. That's kind of depressing for a journal that is under the umbrella of Nature, but whatever. So with that, thanks for listening, and if you have any questions, please leave them down in the description below.